Let's talk now about the glial cells. And the main thing about the glial cells, we said these are supporting cells. So they have different functions, but in general, they are helping the neurons to complete their functions. So they are helping the neurons to perform what they have to do. Based on the location of the nervous system, whether it's the central nervous system or the peripheral nervous system, we have different types of glial cells. For the central nervous system, we have astrocytes, we have oligodendrocytes, we have microglia, and we have ependymal cells. For the peripheral nervous system, we have satellite cells and Schwann cells. Now, astrocytes and satellite cells are providing the same function. Oligodendrocytes and Schwann cells are providing the same function. Whereas macroglia and ependymal cells, they so far we are not aware that they are existing in the peripheral nervous system, and these are their function. Let's go very briefly into each one of these cells. The first one is the astrocyte. The name is coming from their shape. So these are star-shaped cells. We are talking about these cells. And as you can see, these have multiple extensions from them. As you can see, they are connecting to the neuron. This is the neuron. But also they are connecting to other structures, to ependymal cells, for example. Their main function is to support and brace the neurons. So these are for supporting neurons in their functions. Also, they have connections. They can communicate or they connect to blood vessels. And you can think that they can bring some information from the blood vessels to the neurons. The other type, or the second type, is something called oligodendrocytes. The name, it means the cell of a few branches. These are the oligodendrocytes. The main thing about these glial cells that they surround the axons of the neurons and insulate them with myelin. So they insulate the axons with myelin. So this like light blue material, light green material is the myelin sheath. And this material, later maybe we'll talk about it. It helps in faster transmitting of the signals. So it helps in faster transmission of signals. These are the oligodendrocytes. The microglia, these are small cells. They have, we can think of immune functions of the nervous system. So they can ingest and digest pathogens that might cause some diseases. So they clean the nervous system. Lastly, the ependymal cells. These are the ependymal cells. And these are important because they are responsible for filtrating blood to make the cerebrospinal fluid, the CSF. This is something we are going to deal with it later. For now, just understand that their function is to produce the CSF or the cerebrospinal fluid. Let's see the neuroglia of the PNS. So all of the cells we saw earlier, as we said, these they were of the central nervous system. Let's see now the peripheral nervous system. We have two major glial cells. We have the satellite cells and we have the Schwann cells. And if you remember, the satellite cells, very similar functions to the astrocytes. So they provide support, performing similar functions for the astrocytes, mainly supporting. And the Schwann cells, very similar function to the oligodendrocytes. So they insulate axons with myelin sheath in the peripheral nervous system. Now, here, as you can see, this is a cell in the peripheral nervous system. This is unipolar neuron. The satellite cell, they are located on or around the cell body. Now, if we go back and compare it with the astrocytes, as you can see, one astrocyte can contact two or more of the cell bodies or of the neurons. Here, no, we have several satellite cells on one neuron, but they are performing the same function. The same thing with the Schwann cells. With oligodendrocytes, as you can see, this is an oligodendrocyte. It can connect to several axons and insulate them. Okay, same thing here. Several axons and insulate them. Here it's a different story. Here we have the axon, and on the axon there are several Schwann cells. So it's not one cell for several axons. No, here one axon needs several cells. So for the myelin, we have been talking about the myelin sheath. So for the myelin, it's a substance that is very rich in lipid that would surround and insulate, as you can see in the images, that will insulate the axon. This is the axon. So it will insulate the axon. Now, why this is important? As we said earlier, we need faster transmission for the signals. So this would help in a faster transmission of the electrical signal, or technically of the action potential, which is something we will see later. Now, not all axons have the myelin sheath. Some axons have it, we will call them myelinated axons. Some axons, they don't have it, and we will call them non-myelinated axons. And you would expect that the signal or the action potential is slower here comparing to here. And we will understand this later as well. And the space between two Schwann cells in the BNS is called nodes of Reimvar. What does this mean? If we go here, this is space, maybe this is space. These are called nodes of Reimvar, which is something also we are going to talk about later. But one important thing that is also coming out because of the myelin is the color of the nervous tissue. Okay, so here you can see there is something called the gray matter, and there is something called the white matter. White matter because of the abundance of myelin sheath in this area. Okay, in the gray matter, no much myelin sheath. So what is the concept here? In the gray matter, we have cell bodies, and these cell bodies, they will have their axons sent down. Since all of these or most of these axons are myelinated, and myelin is a white material, so this area would appear whiter. That's why we said like there is abundance of myelin sheath into this area. Why? Because all of these axons coming from all of these cell bodies of the neurons, all of these axons are myelinated axons. That's why it's a white matter. Whereas here there is no myelin, or there is less myelin. That's why this is a gray matter. So that's basically the difference between the gray and white matter of the brain. Last thing, and this is regarding some terms that we are going to use later, and these are a little bit confusing, but just like understand the term, and later we will talk about how we can use these terms. Now, here we mentioned that the cell bodies are located within the gray matter, right? These are located in clusters. So each group of cell bodies are sitting together, and usually these clusters of cell bodies, they perform certain functions. So we will have like centers. You know, there is a certain center for speech, center for like hearing, center for dreaming, and so on. So the cell bodies there are located in clusters with each other. In the central nervous system, these clusters are called the nuclei. Okay, in the central nervous system, these clusters are called nuclei, meaning that is in the brain and the spinal cord. However, in the peripheral nervous system, these clusters of cell bodies, we call them ganglia. Like this is, for example, in the BNS, and this is a ganglia. This is a ganglia. Nuclei, this is part of the spinal cord. Nuclei would be somewhere here and there. So that is the difference between the nuclei and the ganglia. Basically, it's the same thing, a cluster of cell bodies, But if they are in the CNS, we are going to call them nuclei. If they are in the BNS, we are going to call them ganglia. So I will stop here. Write your questions in the forum, and once we resume, we will go through the rest of the chapter.